So let's pick up where we left off here. Sadie, can I get you to just turn off that front light? Thank you. All right, so we left off here talking about plant and animal cells having lots of similarities. Okay, they have far more in common than they have that's not in common. All right, obviously the main differences though have to do with how they get their energy. Okay, they're what we call mode of nutrition, which we'll talk about next week. Okay, the mode of nutrition for a plant is that they are photosynthetic. Okay, we call that a photoautotroph. Means light self-feeder. Okay, and animals are what we call chemoheterotrophs, chemical other eaters. Okay, so they have to eat other things basically. All right. So what we're looking at though is that there's a lot in common. Okay, obviously they both have a nucleus. Okay, the big purple thing in the middle. All right, the big purple thing in the middle. What does the nucleus do? It stores DNA. I like that because it's the first time I haven't got brain of the cell as the answer. I hate that answer. Okay, it's not a brain. A brain is made up of many cells, and the nucleus is not a brain. Okay, what it does is it actually it doesn't really control what goes on in the cell, but it's where the cell goes to get the information it needs, because that's where your DNA is. Okay, the nucleus, is really more like a library. All right, and people go there to get information that they require. If the cell needs to make a protein, that's where it goes, because that's where the instructions for making that protein are. If it needs to repair a damaged organelle, it goes there for the instructions on how to do it. All right? So in that way, people get the idea that the nucleus controls what's going on, but in actual fact, most of the parts of the cell work fairly independent of each other. Okay? They're dependent on each other, but they work independently. All right? So obviously, yes, the nucleus is a big deal, okay, because all living things on Earth have DNA, and DNA is the code for doing stuff. Okay, so it's important that we have, at least in these kinds of cells, a place to keep that stuff safe. All right, because if it gets damaged, is that bad? Yeah, if your DNA gets damaged, okay, then certain parts will not work correctly, okay, things like that, and we don't want those kind of things to happen. Okay, we've also got this blue stuff here that you can see in there, right? The blue stuff is what we call endoplasmic reticulum. You will be relieved to know that you only have to call it ER, okay? But it is technically endoplasmic reticulum. And what it does is it's probably one of the busier organelles. Between the rough ER and the smooth ER, okay, because there's two different kinds, right? The rough stuff is this stuff here, and the smooth stuff is this stuff, okay? And there's some more of it over here. You can see that there's lots of it, right? There's some more up here, right? There's a lot of it down in this area down here. It kind of goes everywhere because it's kind of a transportation system for moving things around, but it's also responsible for detoxifying poisons and things like that that are either produced by the cell or that might, be ingested accidentally or on purpose. Okay, things like that. It's the thing that kind of cleans it all up and keeps the cell going. All right, they both have this orange thing, okay, which is called a Golgi apparatus. Golgi apparatus is kind of a garbage man. It gets rid of all the waste products produced by the cell. Okay, they both have these sausage-shaped orange things, okay, throughout their cell. Okay, those are mitochondria. They burn sugar for energy. All right, and we've said that sugar is the fuel. Whether you're a plant or an animal, sugar is your fuel, glucose specifically. Okay, and it's just how you get that glucose that is different. For a plant, they use their chloroplasts. The chloroplasts carry out photosynthesis, carbon dioxide and water and sunlight make glucose, okay, and oxygen. That glucose then goes to here, and after that, everything is the same as it would be in an animal. Okay, an animal ingests food, okay, and then there's organelles called lysosomes that basically break the food down into different types of materials, okay, glucose, fats, right, stuff like that. And then the glucose goes to the mitochondria, gets burned for energy, and after that, all the processes are the same. All right, so there's way more in common than there is different. Right? And that's really the important thing to remember here, because it leads us to the conclusion that there must have been a common ancestor way, way back when life began, let's say down here, right, that led to both types of organisms. All right, everyone follow me on that one? So, yes, very early on in the tree of life, there would have been a branching, okay, of animals and plants. They would have diverged quite early on, right? But obviously, if certain things work, they're kept around, right? And most of these cellular processes are common to all living things on Earth. All right. Now, microscopy. And I forgot to grab a microscope. I'll be right back. Kind of need one to explain this stuff. All right. So when you get a microscope out of the cupboards in the lab, they will come with a dust cover on. That must be removed before you can use the microscope. Okay. Now, the microscopes are expensive, delicate pieces of equipment. And we have had one single problem with them based on their design. As you can see, there's a bunch of electrician's tape around the transformer, okay, on this power supply here. The reason for that is dinky cord, heavy plug, right? Please don't allow the heavy plug to dangle, because if it does, it just comes disconnected either from here or from where it goes into the microscope at the back. All right, so please be careful that the cord is not dragging, dangling, or anything else, because if it's on the floor, I've actually seen people step on the cord, keep walking, and as they walk away, the cord tightens, they pull the cord out, but the tension pulls the microscope out of their hand, and then two things are broken. And then they hear the thumping sound, and then they hear the ambulance sound, and then they don't hear anything after that. Please be careful with these. They're very, very expensive and very, very breakable. All right, so handling techniques. The first thing you might have noticed when I walked in that I was carrying it with two hands. That is also how you will carry it. Okay, with two hands. One on the arm, one on the base. Okay, that's what that's called. That's not a neck. Okay, it's an arm. Okay, so one on the arm, one on the base. Carry it carefully, because if somebody bumps you, okay, because I've seen this happen, someone gets it right on the funny bone while they're carrying this, but they're only carrying it with one hand. Okay, you get hit on the funny bone, and boom, your hand relaxes, and off goes the microscope. Okay, so please always carry it with two hands. All right, when you're moving from place to place. All right, on the revolving nose piece, that's this part right here, okay, you will see that there are three objective lenses, okay, short, medium, and long. They're also known as the scanning power lens, which is the short one with the red band on it, okay, the low power lens, which is the kind of medium one with the yellow band on it, and the high power lens, which is the long one with the blue band on it, okay. Each of them obviously magnifies differently, okay, but you still need to use all three, believe it or not. It is very difficult to locate and focus a specimen by starting on the high power lens, okay, because the high power lens magnifies a great deal, but it magnifies a very, very small area. The scanning power lens, the lowest one, by comparison, doesn't magnify very much, but allows you to see a much greater area. So you want to start always with the scanning power lens, because it'll be easier to find your specimen. You won't see a lot of detail on that one, but at least you'll know where it is. Okay? And then you can start moving up in magnification, seeing more detail because you've actually found the specimen. Now, these are a little better than the ones you had at John Paul II. All right, they have adjustments on the stage that will allow you to move the slide around while you're viewing it without having to use your fingers on the stage. Okay, so what you always want to do when you look through the microscope, okay, I'm just going to put in an extra one here. When you look through the microscope, no matter what magnification you're looking at, you'll see a circle, and you'll see this, a pointer, okay, that points towards the middle of the viewing area. It's actually just a little wire, but when you look through the microscope, it looks a lot bigger than that for some reason, because the microscope magnifies everything. Okay, it is supposed to be there. Right? I've actually had people bring their microscopes up to me and say, Mr. Couder, there's something wrong with my microscope. There's this big black thing in the middle of it. So I'm, oh, that's not good. So I look through the microscope, and this is what I see. I'm like, what black thing? You know, that big black thing that's pointing at the middle. Go back to your desk. Yeah, that's supposed to be there. It's the pointer. Okay, it tells you where the middle of the field of view is. Now, that may not seem all that important, but it is. Because when I look on scanning power, okay, here's what I might see. I might see a bunch of little tiny cells, okay? And they'll be small when I look at them on the scanning power because they're not greatly magnified. What I'll want to do is choose one of those to be the specimen that I'm going to look at. Before I move up, I want to make sure that that particular specimen is on the end of the pointer. by moving the stage around until it gets to there. Okay, then you move up to the next magnification, and lo and behold, it'll still be in the middle. All right, here's why. When I move up to the next magnification, I see greater detail, but unfortunately, I see only this much area. Because I've magnified, I can't see as much space. And so if I don't move my specimen to the center, I won't see it anymore because it'll be outside the field of view. Everyone following me on that? Yes? Okay. Now, again, when you do that, the specimen may move a little bit, and it'll probably be out of focus, and you'll have to refocus it. But you'll see much more detail. Okay? Then, once you've got it back on the end of the pointer and kind of looked at what you need to look at, you'll go on to high power. Well, high power will only allow you to see this much, but of course the specimen will be way, way bigger. Okay? It'll take up the whole field of view now. All right? So, in essence, okay, what you'll see then is, I'll try and be really fancy here. Okay, you'll see, well, that didn't work out very well. Whatever. Go over there. Okay, now let's say that we're looking at that specimen, but now we're on high power. It might look like this. Okay, I've now only magnified that tiny little space I was looking at before, but now, of course, I can see way more detail inside of my specimen. Okay, everybody with me there? All right. On the microscope, in order to focus, there are two different sets of focusing knobs. Okay, there's the big one here that's called the coarse focus knob. Okay, and you can see that when it turns, the stage moves quite a bit. Okay, in fact, you can't even turn it a full turn, all right, before the stage is all the way from the top to the bottom. The other one is this one called the fine focus knob. If you're on high power, this is the one you're going to use. Okay, for two reasons. This one, even the slightest movement, okay, will go right through focus, and you won't even see it happen because it'll happen so fast. The fine focus knob, you see here, I'm cranking it like crazy, and the stage is hardly moving at all. All right, it allows you to make really, really fine adjustments in the focus of the specimen. Okay, the thing you have to remember is when you're looking at a specimen, it's small, yes, but it's still three-dimensional. It has length, it has width, it has depth. Okay, and there are some things that we'll look at where when you initially get them in focus, you'll be focusing on the top of them. And then as you continue to focus, you'll focus through the specimen and you'll see different parts. Okay? It's like if I'm standing here at the front of the room and I'm looking at Samuel, okay, Stephen's out of focus for me. Right? Because he's not the thing I'm focused on. He's too far away. Right? But if I look at Stephen at the back, Samuel goes out of focus. Right? You guys know what I mean? Well, that's what you're doing when you're looking at a specimen under the microscope. You may not see all of the specimen at one time. On some you will because they're that small, right? But on some you won't, and you'll actually have to focus kind of through it in order to see all of the parts. And again, you'll want to do the fine focus knob in order for that when you're on high power. On scanning power, the coarse focus knob is the way to go, okay? Because you'll just crank like crazy with the fine focus knob and not see anything, okay? Other reason you want to use the fine focus knob on the high power lens. See how close the high power lens gets to the stage? Okay, there's a lot of torque on this coarse focus knob, enough that you can actually break a microscope slide with the high power lens if you use the coarse focus knob, and I've seen that happen as well. That also usually generates the ambulance sound. Okay, so because it's not just the slide that gets broken, it usually also scratches the lens, and that's bad because even a tiny scratch on something that magnifies things is really bad, because a tiny little scratch you couldn't see with the naked eye looks like a big jagged canyon when viewed through the microscope. All right, so please be careful with those kinds of things because obviously this stuff is expensive and delicate. All right, so you always use the fine focus knob when you're on the high power lens. Okay, the other two you can use coarse, and with the scanning power you're probably just going to use coarse. Okay, you're with me so far? Okay, to turn them on, there's an on switch at the back. Okay, you plug them in and turn them on, the light comes on. If the light doesn't come on, you usually have to press the surge protector, the little reset button, okay, on the ground faults plugins that we have in the lab. And just press the reset button, usually it comes right back on. Unless Mr. Quadere's forgotten to turn on the power. We'll find that out real fast if that happens, because nobody's microscope will work. Okay, and that's a problem that's easily fixed. Also, when you have this on your table, okay, rather than always, your partner wants to look at it, and people have this tendency to pick it up and then move it over to their partner, there's this cool thing at the top. Okay? It goes all the way around, okay? Or you could just step out of the way and let your partner look, because every time you pick this up and set it down, you're going to ruin the focus, right? Because that jarring effect is going to move the stage enough on high power to take the thing out of focus, and then your partner's going to look at it and go, "What are you, drunk? Like, there's nothing here. I can't see anything." And then there you go, get in a fight, and that's not good. All right? So, yeah, make sure that you leave it, once it's kind of focused, just leave it that way. All right. Questions on that? Okay. All right, so I think that covers, lens paper, right. Okay. If you find that there's dust or somebody's eyelash or something like that on the eyepiece, or maybe there's dust on the lenses, that happens. Okay, there's special paper that we use to clean the lenses, because we know that when we take it out of the container, it has no abrasive particles on it, okay, that would scratch the lens. Things I don't want to see. Okay, if you lick your finger and do this, ugh, okay, gross. All right. People are putting their eyes there, right? Don't do that, right? Secondly, your thumb could have small abrasive particles on it that you picked up from your desk or your pen or whatever that could scratch the lens, okay? That and the slimy crap from your tongue that you licked your finger, yeah, it's just going to make that even worse, all right? Similarly, you don't want to just go with your shirt, okay, or whatever else, because your shirt could still have, okay, abrasive stuff on it. If you ever cleaned the lint trap from your dryer, okay, you'd be amazed what comes out of your clothes, okay, or worse, the trap in your washing machine, okay. If you don't clean that out, it actually smells like a dead body in there, okay, when you actually take it out, because what has it got in it? All the oils and gunk and hair and whatever that was in your clothes, okay, ends up trapped in this thing. I had to clean ours out like a couple months ago. It was awful. Okay, so we don't want to have any of that kind of stuff ending up on the microscope slides or on the lenses, because obviously it'll damage them. All right, so be very careful. There'll be special lens paper. If you need it, just come ask for some and I'll give it to you. All right, and when you, like we said, you start always with the lowest power. That's the scanning power lens, and you move up from there. All right, when you return the microscope, always make sure that you return the microscope either with the blank in viewing position, or preferably the scanning power lens in position. One, it's polite because the next person who uses it will want to start with that anyway. And secondly, most people assume that it is in position, okay, even though they've put a slide in and actually seen that it's not, and then they crank this coarse focus knob and break something. Okay, so please always make sure the scanning power lens is in position when you return the microscope. Okay. All right, this is something you'll have to write down because it isn't in your notes, okay? This is going to be important for how we estimate the size of the things we view under the microscope. On your scanning power lens, which magnifies 40 times, the area of the diameter, of the circle that you can view through the microscope is 4,000 micrometers. Okay, a micrometer is 1,000th of a millimeter. All right, so it is 10 to the minus 6 meters. All right, so quite small. But there are 4,000 of them. That's actually 4 millimeters. So the area, the diameter, sorry, I keep on saying area, the diameter, okay, of the circle you see on scanning power is 4 millimeters across. All right? Would that help you to estimate the size of something you were looking at under there? Sure it would. Okay. How would we estimate the size? Well, if this is 4,000 micrometers across, and what I'm looking at looks like that, approximately how big is it? Yeah, I would say I would even accept 2,000. It's about half. Okay, between, let's say, 1,500 and 2,000 micrometers. It's an estimate. Okay? We're not actually taking measurements here. It's a size estimation. You just want to give a person an idea of how big something is when you have looked at it and shown it to them. All right? The low power lens, okay, is 100 times magnification, and the diameter of it is 1600 micrometers across. So you can see as we increase the magnification, the field of view, okay, the diameter, decreases. Oh, incidentally, this little character here, okay, is mu. That's the Greek letter M. Because obviously we can't use mm because that already stands for millimeters. Micrometers needed an M. Okay, so we borrow from the Greek alphabet. Okay, it's basically a U with tails on it. All right, and in fact, when you do this character on the computer, it actually just looks like a U with one long tail on it. Okay, so that's what it is, mu, micrometers. And then for your high power lens, obviously the diameter of the area that you see, your field of view, is smaller, 400 micrometers, okay, and it magnifies 400 times. So when you look at something through the high power lens, it is 400 times bigger than it actually looks to the naked eye. All right, does that size estimation thing kind of make sense? All right, it's pretty easy. And again, guys, remember, it is, after all, just an estimate. It just needs to be close. All right, if you're looking at something in your high power lens and it takes up 3/4 of the field of view, how big is it? 300, right? 300 micrometers, 3/4 of 400 is 300, right? Okay, so that's essentially how you do it. It's not a difficult calculation, all right? That's the hardest calculation you'll have to do in this unit. Does that make you feel good? All right. All right, so field of view, okay, like we said, scanning power lens, 4,000 micrometers, low power lens, 1,600 micrometers, high power, 400 micrometers. To estimate the size of an object, that's what we just talked about. Okay, you can estimate how many objects would fit across the field of view, divide the field of view by that number, or you can do what we kind of did. Yeah, it looks like about half of 400 is 200. Yeah, either way, okay? It's a size estimate, all right? Estimate the approximate size. All right, I want you guys to answer those questions right now, okay? Just on a sheet of paper quickly. We'll go over them in a minute, and then we're going to start talking about your microscopy lab. Okay, so 3 points of the cell theory. All organisms are made of cells. Okay, second point, what do all cells do? Right, carry out the basic functions of the organism, okay? And third one, where do cells come from? Right, they come from the division of existing cells. That's basically what you need to know. Okay, 4 people considered to be the most important in the development of the cell theory. Well, Hooke, what did he do? Right, he saw the little empty rooms. He discovered cells. Okay, and then who came after that, Quinn? Leeuwenhoek, yes. And what did he do? Right. Okay, and he looked at cells that were living, right? Okay, and then Schleiden and Swan came along, and those two guys essentially proved that everything is made of cells because they looked at tissue samples from all kinds of organisms and found that cells were in all of them. Okay, all right. And third one, on what basis can we deduce that plants and animals may have had a common ancestor? Peter? Yep, similarity in their cell structure as well as not just their structure, but the actions that they carry out, right? Their cell processes are similar as well. Okay, good.